Hey everyone, welcome to lesson 12. We're finally on week 12 of the class, and today we're going to be going over microbial control and antimicrobial drugs. So, microbial control falls into the sterilization, disinfection arena, whereas antimicrobial drugs falls into that same overall system of removing microbes, but from us as living organisms. So for most people, that's going to be you're going on antibiotics for some sort of infection, but it can also be antivirals if you have a really bad case of the flu. You can get Tamiflu early, and then also things like antifungals. So in Arizona, there's valley fever. So that's an example of a fungal infection that you might have to deal with using antimicrobial drugs. And we call them antimicrobials because it's very broad instead of just saying something like antibiotics, even though in layman's terms, that's what most people are going to be talking about. Now, when we talk about microbial control, that's going to fall into some very specific buckets. So there's sterilization, which we talked about before, which is the complete destruction of all the microbial life that is in a system, whether that's on surgical tools or it is on a piece of equipment that is being sent into space to monitor the moons of Jupiter. Whichever you're doing, those two things have to be sterilized. There can't be any microbes on them generally. Then there is disinfection. Disinfection eliminates most microbes on an inanimate surface, but doesn't get rid of all of them. It is not a sterile surface, it's a disinfected surface. So this would be things like your hands right after using some sort of, either washing it with soap and water, or if you're using some sort of alcohol on your hands, that will disinfect it, but is not going to sterilize it. There's also antisepsis. Antisepsis is specific because it's disinfection on living tissue. So this is when your living tissues, you cut your arm open, you want to use some sort of antisepsis on it. So you put Neosporin, let's say, on the open wound, and that is a form of antisepsis. That is different than disinfection specifically in a definitional term. We'll use disinfection a lot just in layman's terms of, oh, scraped your knee or cut your finger open. You need to go disinfect that. But in scientific terms, that's antisepsis. Now, there's also sanitization. This lowers microbial counts to safe public health levels. Sanitization, sanitation systems, stuff like that. So when we're talking about sanitation, that is controlling the overall amount of microbes versus sterilizing all the microbes in an environment. So your drinking water has been sanitized in ways. We add stuff like chlorine and fluoride for multiple different reasons, but one of them is to reduce the microbial burden in the water so it is safe to drink. There is still microbes in the water. If you don't boil the water, you're going to have some live microbes in there, but it is sanitized. It is safe. Then in all of these, there's going to be a difference between cidal and static. And usually we talk about this in terms of the use of antibiotics, so bactericidal versus bacteriostatic antibiotics. Bactericidal is killing of microbes, bacteriostatic is inhibiting their growth. So those are two differentiations that we also need to be aware of. Now, what are our methods of control? Because we have drugs, of course, but there are also many, many other methods that we can utilize for the different subsets here, whether it's sterilization or antisepsis, disinfection, sanitization. One of the most common throughout history has been heat. You're going to use heat to cook off the microbes, cook off the spores, and destroy them all into nature's proteins to the point where it's just you're dealing with the building blocks of life rather than dealing with structures of DNA, RNA, and protein. Dry heat is slow and effective. Moist heat is typically more effective and faster, and that's why autoclaves are a pressurized steaming system that is going to just pressure, steam, heat all together be used to rapidly sterilize things. Cold can also be used, believe it or not, because it will slow microbial growth. You will have this slowing down of the metabolism of microbes, and therefore it can be used as a control method. That's what your fridge is for. That's what your freezer is for. Freeze drying, for instance, is fantastic because it's a good way of preserving foods. So all of that comes from cold. So you have heat and cold working together. Another is this osmotic pressure exchange. So a lot of microbes, they can't grow in high salt environments. They can't grow in high sugar environments. And that's why salting and sugaring foods is very, very common. And drying out foods as well, because when you dry out the food, you take away all that moisture. The microbe needs moisture to live. So at a certain point, it can't get any moisture and it dies off. So smoking of foods, like smoked salmon, for instance, are also really common because you're having that osmotic differential to where if you put enough salt on a microbe, all of the water comes out and they die. Same thing with if they're full of salt and there's no salt around, they will at a certain point start shriveling up and dying themselves. Radiation can also be used, so UV light is used a lot, and then of course ionizing radiation. So this is the kind that's really bad for our cells and you don't want to get any on you, but is really good for sterilization. One thing this is used for is sterilization of medical supplies before it's packaged, and also sterilization of things like medical equipment. Then there is filtration. This is the physical removing of microbes from the environment. So you might have heard of HEPA filters. Those are used in hospitals and many other public spaces such as gyms. And those remove microbes by just catching them on a filter so that they can't get back into the air. And this is the same thing with something like a Brita filter, for instance, that you might use for your water. A lot of microbes will actually get caught in those filters and not be able to get filtered out. So those are the physical methods, but there's a lot of chemical methods of control as well. And this is before we get to antimicrobial drugs. This is just general chemicals that can be used. One, of course, is alcohol. Alcohol is fantastic because it denatures proteins, it disrupts membranes, and it is effective, but not deadly to us because we have a skin barrier. So, using alcohol to sterilize your hands, sterilize, I use that word when it's not actually real. You're actually using antisepsis in the scientific terms. But of course, we use these phrases all the time in layman's terms of, oh, you need to disinfect your hands, you need to clean your hands, you need to sterilize your hands. But knowing those differences is important for the scientific side of things. Again, there's going to be different things. Oxidizing agents such as iodine and chlorine can be used a lot, and these are typically used as antiseptics and disinfectants. So you might have heard of iodine being used before surgery. They clean up the area around you with iodine. Of course, that's after you've already been kind of scrub clean. They put the iodine there so when they do a surgical incision, nothing from your skin is alive to go into that interspace, whatever kind of surgery it is. So that's another example. There's also phenols, and these disrupt membranes and proteins and can be used as disinfectants. Unlike alcohols, these can be bad for our skin. So we use alcohols instead on our skin, but phenols can be used typically to disinfect a surface. There's, of course, a lot of advantages and also some disadvantages to this. One of the large advantages is that these are broad spectrum and they're stable on the shelf and the cost is very low. So the disadvantages is you have tissue toxicity. You have, if you're inhaling these things, it's very bad for you. If someone is to say drink even rubbing alcohol that you have, that's bad enough. But if you're drinking phenols or you're drinking these halogens like iodine, someone accidentally gets into that, drinks some of it, it's a visit to the hospital no matter what. So you have to think about how to regulate these things and structure it in such a way you're using what makes sense in a certain place and time. And that's why iodine is used in the hospital doing surgery and not at home. We're not spreading iodine on our hands at home. It just doesn't make sense. Now we get to the big thing, which is antimicrobial drugs. And these can fall into a couple different categories. There's natural, and these are produced by microbes. These are things like penicillin. So penicillin is produced from a mold, and that mold fights off bacteria. We harvest the penicillin that that mold makes, and then we can, I mean, now it's made using genetic engineering, which we talked about previously, but it could be originally harvested from the mold itself and then used as an antimicrobial drug. There's semi-synthetics, which are this bridge between natural and synthetic drugs. Semi-synthetics are chemically modified natural products that are used in a stage of synthesis, typically for small molecule drugs like antibiotics. So you're going to take something natural, you're going to slightly modify it, and that typically is going to improve it for broader use and evading the resistance that microbes can have. So that is a very important subset of natural slash synthetic. And then there is, of course, fully synthetic compounds. And these are going to be a lot of your modern day medicines, as they are completely 100% synthetically made by chemists in a lab, synthesize synthetic, and they are used out in the real world because they work very effectively. Now, for the history of these antimicrobials, Paul Ehrlich was a really big part of this. He developed the concept of this like magic bullet of using drugs to treat and cure people of various bacterial diseases. The top one that he's known for is a drug that actually treated syphilis. Alexander Fleming discovered penicillin in 1928, and that discovery actually sat on the shelf for a long time. And this is why communication among scientists and amongst the public is very, very important because he discovered it in 1928. But penicillin as a drug wasn't deployed until realistically World War II. That's when we started dropping penicillin with a bunch of crates and caches and giving it to soldiers who were fighting in World War II. And you think about that, that's 10 years later. That's a decade later and a little bit more. So that actually, sadly, was a discovery that happened and then sat on a shelf for a long time because no one knew how much of a breakthrough having this product that fights off microbes really was at the end of the day. Then there's Selman Wakeman or Wakesman, I forget how you pronounce it, but he discovered streptomycin and actually coined the term antibiotic. So he coined that term and then it became used much more broadly. And this, of course, was after Alexander Fleming down the road. Now, they discovered some of the first antibiotic drugs, but there's a lot of factors that go into the efficacy and really influence how we use those drugs in the modern day. So, one thing is selective toxicity. It kills the microbes without hurting us. Antibiotics, you're going to get some of stomach ache, maybe have some indigestion, things like that. But with something like antifungal, because fungus are closer related to us and more like us on a cellular level, you're going to honestly end up really, really sick from a lot of these antifungal drugs, even though they're life-saving. So we have to balance that out. Do you use an antifungal? Do you not use an antifungal? Do you use a topical antifungal? So if you get athlete's foot, for instance, you use a topical antifungal, and if it persists, it gets worse, you end up taking an oral antifungal. And then that actually can cause a lot of issues. It can cause liver toxicity and things like that. So not only is the dosage important, also the route is very important, whether it's topical, whether it's oral, whether it's an injection, whether it's an IV you put in your arm, all of those things matter a lot. And then another thing is your metabolism. Every person has a different metabolism for different types of drugs. I mean, we talked about metabolism a while back. It is incredibly complicated. So there's no good way to actually say, what, based on your height, your weight, your amount of water intake, and the type of food you eat, and your metabolism based on your parents or whatever, genetics. We don't have anything like that to tell us how good the drug is going to work. So we actually have to, in our clinical trials, say, we have a drug. This is the amount that works in all people without causing harm. And that's why clinical trials are so important because maybe someone could take half that and they would be fine. Maybe someone could take 10 times that and they would be fine. But we don't know that. So we just go for the optimal for everyone. We know what works and what is safe, and find that cross point of it works and is safe. And that is why it is very, very important to run things like clinical trials. And another reason it's important to run clinical trials and worry about this effectiveness and the different factors that influence using antimicrobial drugs is drug interactions. So, drug interactions can be very, very dangerous because you have one drug that goes down a certain metabolic pathway and is gunking it up, and you use another drug that also follows that metabolic pathway because there's two things there that need to be processed. You're not processing other things, and it can lead to kidney issues, liver issues, lungs, heart, brain. It can build up in different weird ways and interact in different weird ways that we don't know until we give it to a person. So, clinical trials are insanely important. Now, all of these drugs typically act along a certain set of mechanisms, whether it is for uh, bacteria or it is for viruses or it's for parasites, whatever it is. And this is going to be inhibiting synthesis of a critical portion of the bacteria, the virus, whatever it is. So for bacteria, one of these is inhibiting cell wall synthesis. And the cell wall in the bacteria is detectable with a gram stain. So that's something and why gram stains are so important because that lets us batch our treatments very easily into, well, this is gram negative, this is gram positive. We have a set of drugs that works on gram negative, we have a set of drugs that works on gram positive. So that's how we're going to do that. Then we have things that inhibit protein synthesis, whether this is in a fungus or if it inhibits the protein synthesis of a virus that's using your own cells. These are very, very useful and some of the best drugs that we have. For things that have membranes, again, bacteria, parasites, some viruses have membranes. We have membrane disruptors. We have drugs that go in and actually disrupt that membrane and pull it apart and expose the weakened systems that require that membrane to stay stable so they become unstable and they fall apart. And that's fantastic. We have nucleic acid synthesis inhibitors, and those are going to just say, hey, you're not making the DNA or RNA you want to. We're going to stop that entire process so you can't replicate. That's also fantastic. And that goes into the bacteria static or viral static side versus the cidal side. So if you're disrupting membranes and that's destroying it, that would be cidal. And if you're preventing it from replicating, that's static. The last thing, and this doesn't really work for viruses, but works for the higher order microorganisms, is metabolic pathway inhibitors. So you look at something that is an anaerobic bacteria, it requires that there's no oxygen, and somewhere along its metabolic pathway, it makes oxygen, but that really fast reactive oxygen species is then converted into something else because it doesn't want the oxygen. Well, if we block that pathway, it builds up this reactive oxygen and it dies. So that's another thing that we can use, which is really, really cool. Of course, we have to be worried with all these drugs about drug resistance. So this can come from a different set of mechanisms. Of course, you can block the drug entry into either the cell if it's a virus and you're using a host cell, or the cell if it is a bacteria, a fungus, a parasite. You can pump the drugs out. So these are what we call efflux pumps. You're going to have the drug come in and you go, I don't want that. I want to live. And then you pump it out very, very quickly. There's the enzymatic inactivation of drugs. So a drug comes in and the microbe goes, oh, I'm just going to inactivate that. I'm going to use some sort of enzyme to chomp that in half into two things that, when separated, don't harm me at all. And then I process that and I actually use it as energy. How cool is that? Or, I pump out the excess. There's altering of the drug target. So if you think about using these different like membrane disruptors, well, they target the membrane, but if you alter it, then it doesn't target the membrane anymore. Maybe it targets some sort of extracellular protein that's floating around in the space. And instead of the drug attacking the microbe, the drug just attaches to something and then floats away harmlessly, both to it and the parasite or bacteria and the host. So they just are going to alter the drug and it's going to get rid of it. And the last thing, and it kind of goes with that drug target altering, instead of altering the drug and what's targeting, you can alter the target of the drug. So you can say, wow, our our specific protein that we need to get rid of those reactive oxygen species keeps getting mucked up. We're going to pump out some really fast, messed up ones that look the same, but aren't quite the same. And in us, they do nothing, but the drugs prefer to bind to them. So we're going to produce a ton of those, and the drug comes along and it binds to that, and then that drug no longer does anything. So these are all ways that microbes can avoid being drugged with what we have already and build up resistances over time because none of that has a choice from the microbe, right? The virus isn't making that choice. It's just somewhere along the evolutionary line. There's a mutation. The mutation serves that purpose. And all of a sudden, we have bacteria that are resistant to penicillin. That's a huge, huge problem is bacteria that are resistant to one of our still best antimicrobial drugs. Well, in this case, antibacterial drugs, which is penicillin. And you will typically no longer get penicillin pills. If you're getting penicillin as a treatment, it's a big shot. It's like 6 milliliters of penicillin. It's a huge amount. It is not a fun time for sure. And you'll get that shot. And the reason that you do the shot now is because people don't take half the penicillin thinking they're better, get sick again, and now we have another microbe that's resistant to penicillin. That's a huge problem because there's some microbes that really penicillin in that family of antibacterials is the only thing that works. So once they break through, it's kind of over, and they now will be really difficult to treat over a course of time. Now, we're getting into the causes of what causes drug resistance. One thing is overuse. You give a million people a course of antibiotics, and even if 900,999 of those people take it, the small portion who don't can trigger bacterial resistance. And then they're still sick, and that bacteria spreads and it spreads and it spreads. And that's why if you get antibiotics, it always says take the full course. Even if you feel better the next day, you have to take that full course because it can come back and it can be very, very worrisome with very deadly microbes that when they do come back, you might go to the doctor and the doctor says, well, we have one more thing we can try, but after that, that's it. And usually there are a couple lines to these antimicrobials, and there's infectious disease doctors in the hospitals that are making the decision on these final lines of defense and who they can be used on in what context. And they're typically only used in the hospital because it's very serious when these are deployed, and you have to make sure that people are taking all their antibiotics, or you can get this antibiotic resistance from these patients who don't finish their treatment. And that's the incomplete antibiotic course. It's talked about a lot in public health, and it is a huge issue. The other thing is misuse. If you say, well, I've been sick for 4 days, I have a head cold, the doctor might not give you antibiotics. They might say, if it goes away in a week, you're fine. And if it doesn't go away in a week, come back. And the reason they say that isn't because they want to prolong your suffering. It's because they don't want to give you antibiotics and wipe out your microbiome and have you feel sick, even more sick than you already do, if it's a viral infection. Because if it's a virus, a lot of the times after a week, it's gone because our bodies are really good at fighting viruses. So that leads to this kind of annoying thing where I have definitely gone in and said, I've been sick for a week. Can I have antibiotics? And they go, wait 3 or 4 more days. And I come back. I'm like, it would have been really nice to get antibiotics 3 or 4 days ago. And now I'm sicker. But I understand that the reason they do that is they're trying to prevent this resistance from building up. And some examples of bacteria that this has already happened to and is a very, very big public health issue is MRSA. People get MRSA infections from unwashed gym equipment and just cutting themselves open on, a random piece of metal that's sticking out from a display case in the store. And because someone else has bumped into that, cut themselves, and infected it with MRSA, they then get MRSA and they go to the hospital, and the hospital says, this is an antibiotic resistant MRSA. We can't do much for you. We're going to put you on supportive care. We're going to give you what we can, but it is a deadly disease in these cases where it is antibiotic resistant. We also have multi-drug resistant tuberculosis. Tuberculosis is a horrible disease. It's killed a lot of people throughout time, and we have multiple drugs for it, but in a lot of places that have a lot of tuberculosis, they're not getting the right drugs or they're not finishing their drugs. and it's not even their fault a lot of the times. They can't get the whole course of drugs they need because tuberculosis treatment happens over the course of months instead of a week with a lot of antibiotics. So you run into these issues with access to healthcare, access to treatment, timing of treatment worldwide that has led to multi-drug resistant tuberculosis, where if you get it, you gotta hope you're healthy. If you're old, if you're immunocompromised, if you're a baby, which I know no one watching this is, but if you are, in that situation, you are highly likely to have either a lot of complications or maybe die due to that infection. So that's why controlling the use of antimicrobial drugs is important. That's why trying to have really good healthcare systems is important and is kind of what I want to leave you with. That while we're constantly investigating and coming up with new drugs, whether it's for cancer, whether it's for different immunologic conditions like rheumatoid arthritis, whether it's for MS, whether it's for multi-drug resistant tuberculosis. These are all being developed, but we have a lot of good treatments now in that antimicrobial space, and we like to keep them as functional as possible as a global community of scientists, of medical personnel. And that's why really honing into and making sure we're not overusing them, we're not misusing them is so important. So I will leave it there, kind of on a somber note, and just say we covered everything as far as how we can control microbes and deal with antimicrobial drugs and their resistances. Hopefully, this is something that always remains a fact of life, but not a overpowering fact of life where you hear about in headlines all the time. And we can continue to coexist with all these microorganisms very, very well. And when we kind of get overrun with them, there is an option for us at the end of the day that just comes from the pharmacist, and we take a week of pills, and then that's it. So, hope you enjoyed the lesson. Hope you have a fantastic week. And of course, let me know if you have any questions.